we see that these vector-borne diseases, the fact that they have to go through a vector is also something of a potential weak link that we can target. If we can block the vector or block transmission in the vector, we can block the disease. And so I think everyone's sort of on the same page with that and realizes how important vector control is. Now, how do we do it? One way is just to kill the vector, which is difficult. And I think we all know that vectors do evolve resistance very quickly, but it's a very important strategy to try and kill the vector. I think that there might be some other ways of doing it also that don't necessarily involve the vector. And I think that's kind of the line of research that I've been pursuing. In this slide, this is just sort of a cartoon, but I've labeled some mosquitoes in red and some mosquitoes in blue. And the idea is that when you look at a population of Anopheles gambiae, for example, they're not all the same. They're not all equivalent with respect to their ability to transmit diseases. And I think that there are already some biological phenomenons that prevent some mosquitoes from transmitting disease in nature. And so I think that's what we're trying to investigate further, to understand whether we can use some of these to prevent disease transmission. And so, of course, this is something which people have been speaking about for many years. It's not necessarily something new, but when you look at these mosquitoes in the wild that can't transmit diseases, what is it about them, and how can we exploit that? So we know that mosquitoes are genetically different. Some of them are naturally resistant due to their genes, and that is something that can be investigated to create mosquitoes that genetically are resistant to diseases, which is very interesting. The environment plays a key role. So the temperature at which mosquitoes develop, their nutritional, their access to nutrition affects their ability as vectors. So there are non-biological factors in the environment that also affect it. But then there's also the microbiome, and that's really the thing that we focused on a lot. So whether mosquitoes have certain microbes can affect their ability to transmit diseases. And so we think that, for me, that seems like the best bet for finding something that we can actually use, because we know microbes are infectious. And so if we can spread microbes that block transmission, we could prevent that cycle of the disease. So that's all well and good as a theory, but what about trying to actually put this into practice? So the first thing that we did when I started this work now, probably about 6 years ago, was to say, okay, what is naturally in mosquitoes, and are any of them potentially useful, things that are already there? And of course, this isn't something new. There's been a lot of work on microbes associated with mosquitoes even before I started researching it myself. But most of the work actually focused on bacteria. So we know quite a lot about the bacteria that live in mosquitoes. And there are indeed some quite interesting ones. But what's less known is about the fungi and also eukaryotes that are associated with mosquitoes. So that was kind of an area that we thought we would focus on a bit. As you can see on the graph, we found, when we just sort of did a wide survey, we found lots of different fungi associated with mosquitoes. Some of them had been found before, some of them not. One that sort of jumped out at us was this Microsporidia, because it wasn't found in all mosquitoes, but when it was present, almost all of the microbial content of that mosquito was Microsporidia. And it was actually in quite a high number of mosquitoes, not insignificant. In the first sort of studies, around 10%. So we thought, okay, this has some interesting characteristics. Let's see whether it has any effect on the biology of mosquitoes. Okay, so what is this Microsporidia we actually, this Microsporidia MB, we gave it that name because it hadn't actually been described before. It was related to microsporidias that are found in midges in Siberia. So somehow linked to dipterans, but something rather novel that had never been described in mosquitoes before. We found, we looked for it in a number of different sites in Kenya, and it seemed to be very widespread geographically. So all the locations we sampled actually had this Microsporidia. And we started to look as to whether we found it with Plasmodium. And so the rates of Plasmodium were a bit too low to prove that it could be inhibiting, but we did never find Microsporidia and Plasmodium in the same mosquito in the wild. So that gave us the first sort of indication that maybe this was having some effect on vectoral capacity. In some of the more, in some of the sites where we collected more regularly, we realized that the levels of Microsporidia actually were going up and down quite a lot. And in some seasons and in some sites, we're getting quite a lot of Microsporidia. So in this case, we were getting up to 20%, sometimes 30%. So a lot of mosquitoes were actually infected, and that was quite interesting. The fluctuations also were interesting and seemed to be happening after the rains. We got an increase in Microsporidia. We brought these Microsporidia infected mosquitoes into the lab, and we started to use microscopy to look at where the Microsporidia was. Something that struck us initially was that this Microsporidia infection was very localized, and we found interesting infections in the ovaries of mosquitoes. And we also found a lot of it in the gut, but the pictures I'm showing you here are a mosquito ovary, and you can see the morphology of the Microsporidia in there. We then set up experiments to test whether there was actually a transmission blocking effect on Plasmodium. So I mentioned that Microsporidia is in the ovary. It's also transmitted from mother mosquito to offspring mosquitoes. And so that's a very interesting characteristic, and it allowed us to collect mosquitoes from the field that were infected, let them lay eggs, and then take those F1 mosquitoes and challenge them with blood that had Plasmodium in it and see whether the ones with Microsporidia could ever take it up. And so that's basically what I'm showing you here, is that we get mothers that are infected, the darker dots are the infected ones. We then have lots more offspring that are also infected. We then would pool these, let them feed on blood, and test whether they become infected. And so this was quite a striking result. And the data that we got is shown on this slide, is that the Microsporidia infected mosquitoes would never take up the Plasmodium parasite, suggesting that there's a really strong transmission blocking effect. So that was really the sort of key starting point where we thought, okay, this could have some potential for utility. We could maybe use this in some way, but of course there are challenges with trying to get enough mosquitoes infected that this would actually block transmission on a sort of larger scale. And that comes to sort of the next phase of the work is, okay, well, how could this spread through a population? What we wanted to know was, is this hurting mosquitoes or not? And so we looked at infected mosquitoes. Are they able to lay as many eggs as uninfected mosquitoes? And actually there was no difference. So it doesn't seem to be hurting them, at least in this particular experiment. Another experiment to look at fitness was the length of survival of mosquitoes, and we also found no difference in the levels of survival with Microsporidia and not. So we seem to have something that's a symbiont, not a pathogen, and is in some ways helping the mosquito because it's making it more resistant to infections. At the moment, we only know that it makes the mosquito more resistant to Plasmodium infection, but it may be making mosquitoes more resistant to other infections. How does it work? We don't actually know yet, but we've got some interesting data that suggests mosquito immune responses are activated by the presence of Microsporidia. And so we did some RNA sequencing experiments, and we found a lot of genes upregulated. Interestingly, a lot of these genes have been implicated in making mosquitoes resistant to Plasmodium. So it's very suggestive, but we're still trying to figure out exactly how it works. So that's all well and good. We've got a transmission blocking microbe. It's transmitted from mother to offspring. Now, how does it actually spread? Because going from mother to offspring makes the number of mosquitoes, it doesn't sort of spread it through a population. We would need to either have it increase the fitness of mosquitoes, and we haven't really found evidence for that. We just found evidence that it doesn't decrease the fitness. But for it to really spread, it has to have other modes of transmission. And we did a lot of experiments trying to see whether we could infect larval mosquitoes, and that never actually worked. So we didn't get any transmission from one individual to another when we put larvae together. But when we put infected adults together, we did get some transmission. So we thought, okay, adults can infect other adults. How is that working? We did a number of experiments where we put males and females in cages, infected males, uninfected females, and vice versa, and also all mosquitoes of the same sex. And what we found is that only when you have infected of one sex, uninfected of another, do you get transmission. So I think that's very strong evidence that this symbiont is also transmitted sexually. So that's basically where I'd like to wrap up. That's kind of where we are with this. There are a number of experiments that we'd like to do now to see if we can develop a strategy to increase the levels of Microsporidia. As I said, naturally they are relatively high in some places, but in some places much lower. If we can increase the levels of infection of Microsporidia, then we believe that that would decrease the rate of transmission of Plasmodium, and that this could actually be a useful symbiont. But there's sort of a challenge there that we have to overcome, is how do we increase the levels of Microsporidia in a manner that's cost effective and sustainable. So I think I'll end there. I hope I haven't gone too far over my time. Just to acknowledge everyone who's worked on this. It's really been very much of a team effort, but I really would like to acknowledge the students that did most of the work, and they're listed there: Lillian, Enoch, Edward, and Godfrey, who have been working very, very hard on this project. But we've also had many other collaborators and funders. Thank you.
Thank you, Jeremy, for that riveting presentation. I'd like to ask the attendees to please, if you have any burning questions, please put them down at the chat area so that when we're done with all the presentations, then if time allows, we can field a few to the presenters. Next up, we have Professor Rosemary Sang, who is going to be speaking to us on the title, Rethinking New Approaches for Vector-Borne Disease Control. On recent disease trends indicative of rising arbovirus transmission in Africa, a cause for concern. Rosemary, take it up. I hope you can.
You guide me how to share my slide.
There should be a share screen icon down at the bottom of your screen. Right in the middle at the bottom.
Yes.
Did you see it? A share screen icon.
Okay.
Yes.
You see it?
Yes, we can see it, and you can go straight to the presentation mode.
Am I on presentation mode?
No, not yet. Yes, now you're on presentation mode. Thank you.
But
you're good to go.
Part of the screen is occupied by the.
Okay, we are seeing
part
it.
of the screen is covered by the presenter. What?
I think you can minimize it. Although from our side, we just see your slides. I don't know if it's affecting you. There should be a way to minimize that.
Okay.
Okay. Did you figure that out?
No. Let me continue and see if it will. Yeah, it will affect.
Okay. So what are you saying is affecting your slides exactly?
The panel where the speakers are.
You can actually minimize it. So there should be a minimize button at the top left or right. Let me check one of them.
Okay.
Yeah.
So the slides are visible. They're okay.
Yeah, for us it's okay, but it looks like on your side, because you have the participants panel on.
No, now it's okay. Now
okay,
it's okay.
Okay, we're good to go.
Fine.
Then. All right, thank you.
Yeah.
Please take it up. All right.
So first I want to thank the organizers in PAMCA for inviting me to give this talk. The title is, I'm going to talk briefly about what we have found out and what we know about disease trends in Africa caused by arboviruses, and whether we should be concerned and what the way forward should be. So just for an introduction, these arboviruses are by now known to many people. They constitute a very diverse group of viruses that are transmitted by mosquitoes, ticks, sandflies, and even culicoides. We are beginning to see viruses being transmitted by culicoides. They are associated with outbreaks and often leading to high morbidity, some mortalities, in some cases quite high mortality, and often this leads to public health anxiety and uncertainty. And when you have that situation, then the ministry faces pressure even from the media houses, putting pressure on the government to take action because of the impact of these viruses in populations. They present challenges, significant challenges in terms of being able to detect transmission when it is occurring within a population or when there's an outbreak going on. And usually that takes time to know what's going on, even as the disease takes root. And even management is a problem because, as we all know, it's very difficult usually to find treatment for viral infections. And prevention, which should be something for PAMCA to consider seriously. All of us in PAMCA need to think seriously about prevention options. And in most cases, what is available right now is very limiting. So because of the pressure that often arises from these outbreaks, this puts a pressure on rural health facilities, which cannot manage to handle many seriously sick patients. So you find that the health system in such rural places, which is already weak, suffers greatly. And therefore, the importance of these infections. How big is this problem? We are aware by now that there's over 600 arboviruses that are listed in the International Arbovirus Catalog. And over 160 of these are known to cause disease, either in humans or in animals. And when I started working with arboviruses, these viruses were The catalog had, I think, about 550, but the numbers are growing steadily because people are identifying viruses as time goes on, as research increases. And all of them are associated with vectors which transmit. They are found in ecosystems circulating amongst reservoirs and incidental susceptible hosts. They are very diverse. If you're working with arboviruses, then you have to deal with so many different viruses belonging to different families, and this can be quite challenging. It's hard enough, as we already know, dealing with one virus like corona. So you imagine having to try to understand all these virus families that are very diverse. And what we have done to try and find out which of these viruses are circulating in our ecosystem is mainly through sustained surveillance. Over time, researchers have accumulated data, and which has shown that many viruses belonging to all these different families are circulating in countries in Africa. And these are hiding or sitting somewhere, circulating amongst vectors and reservoir hosts or animals or humans, with potential to emerge later on as public health threats. And therefore, it's important not to ignore them. It's important for us as vector scientists to pay attention to them. Sustained surveillance has shown circulation of known and novel viruses. I want to start with some of the ones that we have seen recently through surveillance in Kemri and also in Isipe that are associated with sandflies. Sandflies have been ignored for long, not considered as important vectors of arboviruses, but it is important that we begin to focus on them, even as we are having outbreaks of leishmaniasis out there. It could be that there is leishmaniasis, but there could also be something else going on, because all these are occurring in the ecosystem. There was an outbreak of sandfly fever virus in Ethiopia in 2011, and after it ended, there hasn't been much going on, but surveillance is picking up, and people are beginning to see more sandfly-borne viruses that could be causing fevers out there that we are not aware of. Then we have tick-borne viruses. There are many tick-borne viruses circulating. We have seen many different viruses circulating amongst ticks in our tick-based surveillance, including one virus called Crimean Congo hemorrhagic fever virus that has been reported in many places. We have seen it, of course, here in Kenya. Uganda is the latest to report outbreak of Crimean Congo. And I think they have had outbreak even in 2018. South Africa has had multiple outbreaks, and West Africa too, including Senegal and Mauritania are experiencing outbreaks. So this is an important virus, but because of limited surveillance, there isn't adequate data being detected on human cases that are occurring. There are few human cases being seen, and yet the virus seems to be circulating out there among the ticks. And when we do serosurveillance, we see a lot of evidence of previous exposure. There's a lot of antibodies circulating, especially amongst pastoral communities. You find very high seroprevalence of Crimean Congo. So then you wonder, how is it that the virus is circulating in the population and you don't hear huge outbreaks such as the ones that are reported in Eastern Europe and Asia, which cause massive outbreaks and sometimes high deaths. Crimean Congo hemorrhagic fever has a 40% mortality rate for people who are infected. So there is science there to be explored to find out what kind of virus we are having in Africa. And it's important to also continue sustained surveillance so that we map out the vectors. We know what has been done out there. There is little evidence about which vectors are causing the problem in Africa. So rather than wait until we have a big problem, I think it's important for us to continue to do surveillance and to find out which tick vectors are doing the transmission. Then we know, of course, the big epidemic-prone mosquito-borne arboviruses, yellow fever, Rift Valley fever, dengue, chikungunya, and Zika. And when we look at the map that I have projected on this slide, we see that much of East Africa, Central Africa, has got most of these viruses. In fact, in Uganda, which is a little dot in the middle here, is experiencing a disease from all the 5. Of course, it is assumed that we don't have Zika in the rest of the countries, but that may not necessarily be true. So these epidemic-prone viruses are continuing to, they are increasingly causing outbreaks. Even yellow fever that you would expect should be under control because we have had a safe, efficacious yellow fever vaccine since 1937, and it was used to control yellow fever in the Americas. We are still having outbreaks of yellow fever in Africa to this day. And you can see the data, over 100,000 cases with 44 deaths annually. Most of these are occurring in West Africa. They have frequent outbreaks in West Africa. But we are also beginning to see outbreaks in East Africa. And Kenya suffered the first one in 40 years, in 1992. And similarly, Uganda has been experiencing yellow fever outbreaks, even as recently as 2018. And the first one came in 2011 after 40 years. Similarly, in Angola, we had a huge outbreak that took so long even to identify or detect, and continued for a very long time in 2016, with spread into DR Congo. Here in Kenya, we were able to detect travelers who arrived with yellow fever. So the risk of yellow fever in Africa is, and even moving into East Africa, is very real, and something that requires more attention. Of course, the use of the vaccine brings down the outbreak, but what happens in between outbreaks? What are we doing as entomologists to understand this virus better and its transmission? And what can we do about its controlling it, especially in ecosystems where this is being transmitted in sylvatic habitats by sylvatic vectors circulating amongst primates, and from time to time spilling over into human populations. That's the challenge for us as PAMCA. In 2010, the WHO mapped out Africa and identified risk areas, including much of East Africa, falling into high-risk areas for yellow fever transmission. And when this impacted on our tourist travel, that's the time we became concerned. But I think we need to be more concerned about the public health impact it can have. Dengue and chikungunya is continuing to cause problems. It is spreading across much of Africa. Although initially it was thought that it was a problem of the Asian continent and the Americas, it has now hit in Africa. We are experiencing more outbreaks even here in Kenya frequently. They're experiencing outbreaks in West Africa, and the problem seems to be increasing. it has been seen to be mainly associated with high temperatures, human mobility, urbanization, big population growth. So as African population increases and urbanization grows, and often it grows with poor provision of sanitary services, and that is just perfect condition for dengue and chikungunya to spread. So the challenge is upon us. The disease is here and it is increasing. We are in the surveillance that we continually do at KEMRI, we are seeing dengue increasing, especially in the coast, after every rainy season. So, and that then poses another danger of, as the viruses, multiple serotypes increase, we will begin to see more severe dengue going forward. Zika virus is another virus that should concern us. It was declared a public health emergency of international concern by WHO in 2016. At that time, the virus outbreaks were escalating, going to coincide with the Olympic Games. And somehow things have quietened down, and we should be aware that the virus has not gone anywhere. The virus is with us. It originated from Africa, and it is right here with us. We have done a bit of survey at the western border of Kenya and seen that the population at the border with Uganda is exhibiting evidence of exposure to Zika. So I don't think it is just confined to Uganda where it was found. It is present in West Africa. We just need to be cognizant of the fact that it is a threat that will require our attention. And evidence continues to show that although the virus is transmitted by, it has been branded that it is transmitted by Aedes aegypti, but I think there is a big debate about the role of Culex, knowing that Zika virus is actually much more closely related to a virus called West Nile, which is transmitted by Culex species. So there's still a debate about its transmission, and our approach to control may have to be through vector control. Rift Valley fever is another big virus that we have to be worried about, and Kenya alone suffered over 750 deaths in just 2 outbreaks, and that is a lot of lives lost. The virus occurs every 5 to 10 years, and the challenge with Rift Valley fever is trying to control it when the outbreak has taken off. You imagine that there's over 45 species of mosquitoes that are associated with Rift Valley fever transmission. And in some countries, we don't even know, or there is poor understanding of how that transmission happens. From work that we have done in the outbreak in Kenya, in 2006, we did a huge study on the vectors that caused the outbreak. We have seen that even within one country, the transmission is complex, and it involves different sets of mosquito species in different ecosystems. So that puts vector control challenge, it makes vector control a real big challenge for us. And considering that when you have a massive flooding in a terrain like that, that photograph was taken in Garissa, then you wonder how you're going to control mosquitoes in such a massive, hard terrain to traverse. So the challenge is big, and it is for us to be more creative and to think how we are going to address these challenges. So vector control for dengue, chikungunya, yellow fever, Zika, we could decide to target Aedes aegypti because we have known the breeding sites and habitats are well characterized in many places. And we could decide to just target that. But we have to bear in mind also that yellow fever and Zika are transmitted by sylvatic and peridomestic Stegomyia species that may not be amenable to known Aedes aegypti control. So we need to expand scope in the search for vector control tools and to target, possibly target all important Stegomyia species as we go forward, as we become more innovative. The young people are now more innovative. We need to widen our scope and target more species so that we can address the disease in the African environment. I asked a question at the end of my presentation. Is it feasible to attempt to control all arbovirus diseases targeting vector control? The diversity of the viruses associated and the associated vectors is huge and presents a major challenge. So I don't think there is a single bullet for any of the diseases, as we all know. Aedes-borne diseases can be targeted using multiple tools that are well thought out in an IVM approach. And there's need for a lot of creativity to try and find solutions to some of these important epidemic-prone viruses that are spreading and increasing in magnitude in Africa. I know progress has been made towards Aedes aegypti control in dengue endemic countries. So because these viruses were not so common in Africa, we are yet to take off in this respect. It's good that Jeremy is exploring the metagenomics of mosquito agents that could block some of these agents. And my suggestion is to explore more widely and include other species that present a danger to us. So the task is huge, and the challenge, I throw the challenge to all of us to face the arbovirus disease problem. Thank you very much for listening to me.
Thank you, Professor Rosemary, for that insightful presentation. Before I invite Damaris to invite the next speaker, I would just like to repeat what I already mentioned to the participants, that you could put any questions that you have to the presenter down on the chat box, and then we'll get to them at the end of the presentation. So Damaris, please take it.
Yeah, thanks, Professor Rosemary. Thanks, Elijah. We'd like to invite our next speaker, Dr. Eric Ochomo. Dr. Eric Ochomo is a senior scientist at Cambridge and is going to talk to us, present on a snapshot of new approaches to malaria vector control. Welcome, Dr. Ochomo.
Thanks a lot, Damaris. I will proceed to share my screen. Just a moment. Interesting technology sometimes. Okay.
Yeah.
All right. So I'm just going to get into the presentation mode. Great. All right, I was going to say good evening, then I noticed everybody is from everywhere. So hello, everybody. I hope you're keeping well and safe. Today, my presentation is going to be quite a summary of everything that's going on, nothing specific to a study. I just want us to quickly review what sort of approaches there are to vector control, what improvements there have been, basically take this day to reflect on how far back we've come and where we're going. So the title of the presentation is a snapshot of new approaches to malaria vector control. My name is Eric. I work for the Kenya Medical Research Institute at the Center for Global Health Research based in Kisumu in Western Kenya. So just a brief overview. This is data from the 2018 World Malaria Report, and at that point, we were still talking about 219 million cases and about 405,000 deaths. The number of deaths as at last year came down to about 385, though in terms of the cases reported, that remained pretty stagnant. Now, the one thing I'd like us to pay attention to is the fact that a lot of this burden is still in the WHO Africa region, so that is in sub-Saharan Africa. And I've just included a picture of a child suffering from malaria in one of our hospitals in Siaya in Western Kenya. So in terms of distribution, looking at malaria incidence per 1,000 population at risk, this is in 2015, this is World Bank data. We do see that a lot of the malaria, actually more than 90% of the malaria, is concentrated in the WHO Afro region. And looking at Kenya, so when you present this data in terms of a global scale, you think about the country and think about it in terms of the entire country being endemic for malaria or having uniform distribution of malaria. And that's normally not the case. So looking at Kenya, for example, I did include this map here. We see that a large proportion to the country has quite different prevalence of malaria depending on where you look. So a lot of the malaria burden is in Western Kenya, and that is where we are. And in terms of malaria control, vector control has been the main tool that has actually brought down malaria. Between 2000 and 2015, a lot of the reduction in malaria can actually be attributed to vector control. Vector control being indoor residual spraying. As you can see, this is a picture of my colleague Bernard Bongo. Of course, he's all masked, so you can't really see his face. But then basically, this is indoor residual spraying where you have tins with insecticides and they spray it onto the wall so that when mosquitoes rest on the walls of houses, they take up a lethal dose and that kills them. Or bed net distribution. This is again my colleague Brian giving out nets in CIA. And so bed nets and IRS have actually accounted for 79% of the decline of malaria between 2000 and 2015. So other malaria control methods, prompt diagnosis of malaria, of course now using RDTs and microscopy, and effective treatment of all the cases we find with ACTs. And that's really is what has helped reduce the burden of malaria to what we now see it as. Now, the question is it sufficient? Do we still have people coming down with malaria? So in a place like Western Kenya where I work, the prevalence of malaria is between 30 and 40% depending on season and depending on where you are. Of course, it means that we're not doing enough. A lot more needs to happen. And given that, as I've mentioned earlier, a lot of the reduction that has been achieved has been because of mosquito control. We need to figure out new ways to control malaria vectors, the mosquitoes. Now, this slide here, this is a slide where we sort of present data that was reported in the WHO malaria report last year. And basically, it talked about declining incidence between 2010 and 2018. But then the one thing it stated was the fact that between 2014 and 2018, there was low rate of change in incidence. In fact, for many parts of Africa, there was literally stagnation. There is no change year after year, despite continuous investment of millions of dollars into malaria control. Lots of reasons have been given, some of the reasons being insecticide resistance, so that mosquitoes are no longer responding to the dosage of the insecticide on the nets or on the walls. Changes in vector behavior. So for example, mosquitoes that used to bite indoors start biting outdoors. Reemergence of effective vectors. Actually, we do have a couple of papers from a few years ago where we show the reemergence of a notorious vector called Anopheles fenestus, which after the distribution of bed nets sort of disappeared. And then now we're starting to see it again. And then of course, the big question here is outdoor transmission. What if mosquitoes are actually transmitting malaria outside of the house or outside of the bed net area or outside of, outside of the areas where we currently have tools? The picture I present here is a picture of a WHO tube bioassay test, and these are mosquitoes that are literally, they are surviving exposure to a bioassay. This was 2 hours after they'd been put in the tube with insecticide, and they're still standing there. So insecticide resistance, literally. And then down here, I present some data from actually work we're currently doing in Western Kenya, where we look at the distribution of mosquitoes inside of the house, outside of the house. Anopheles gambia and Anopheles fenestus are main vectors. Anopheles custani is what's called a secondary vector. It's really not been implicated in a lot of transmission. And so we're just looking at the distribution of mosquitoes. Now, if this was 20 years ago in the 90s, your fenesters would only be inside the house. All your Gambi would be inside the house. Right now, the scenario is changing. So we're starting to see a lot more Gambi outside the house. We're seeing some fenesters shifting to being outside of the house. Now, does that mean they're biting outside the house? Does that mean they're transmitting malaria outside the house? All questions for us. I mean, as PAMCA, how do we, what can we do to address these concerns? And so here, this is a not very complicated diagram presented by Jerry Killen in 2017. And this basically just shows what aspects of mosquito behavior we can exploit to control them. So starting at the bottom, this is daytime. During the day, what are mosquitoes doing? They're basically resting outdoors and waiting for the eggs to mature so that they can go and lay them. And then at dusk or at dawn, they start moving. So when they move, they're either moving to look for cattle outdoors, or they're moving to look for humans that are sleeping outside of houses, or they're outside foraging for sugar, looking for plants where they can feed on sugar. So they could be doing any number of factors. And then in the nighttime or early in the evening, they're swarming. So they're mating at that time. And that's the same time when the gravid females are laying their eggs in water. And then also in the night, they're looking for human hosts. So that's the time when they come into the house. So when they come into the house, depending on whether or not you have a bed net, they may come in, find a bed net, and go out again to seek alternative hosts. Or they could come in, find you outside of the protection of a bed net, and therefore be able to feed. And so, given this description of the behavior of the mosquito, we're actually able to figure out what additional tools we can bring into the malaria control, vector control space. And so currently, the specific behaviors that we're targeting for vector control are sugar feeding, the fact that they consistently swarm in the same manner. So mosquitoes tend to swarm in the same general area every single evening. And so now that we have that information, what can we do with it? Mosquitoes have multiple hosts, so they feed on cattle, they feed on humans, they feed on pigs, chicken, literally everything that you have around your home. Chances are mosquitoes will feed on it. They come into the houses for blood meals, and of course that is why IRS and the bed nets have been very effective. And then there's also sustained night feeding. So despite all of these controls, we still see that they prefer to feed at night. So what can we take advantage of all these behaviors in a bid to be able to control them? So here I present a slide showing what's called a spatial repellent. So this is a spatial repellent device. This is a device that was tried out in 2 sites. One was in Peru, and the other was in Indonesia, in a previous study that was funded by Bill and Melinda Gates Foundation. In Indonesia, the trial was trying to look at the impact of spatial repellents on reduction of malaria incidents in children. And then in Peru, it was looking at the impact of spatial repellents in reducing vector-borne, arboviral diseases, so dengue fever or other hemorrhagic fevers that are transmitted by mosquitoes. Now, there has already been the paper describing the results of the Indonesian trial have already been published, and it was pretty. So the results were did not show a strong effect, mostly because Indonesia has very low transmission, and so for you to achieve to be able to show impact in a trial like this, you conduct what's called a randomized control trial, where you identify 30 clusters, the size of a village. So 30 of them, up to 30 or 60 of them. So 30 of them you give the product of interest, 30 of them you don't give anything, and you monitor malaria in this population for about 2 years, 3 years, to try and see if you constantly have a reduction in malaria incidence in the area where you've given the product of interest versus the area where you haven't. And so the trial in Indonesia did not come to conclusive results because the malaria incidences there were already pretty low before they started. So it was very hard to show anything. And so currently, United is funding, through the University of Notre Dame, is actually funding a couple of sites, including our site in Kenya. And we will be evaluating spatial repellents in Busia, hopefully as soon as we can figure out this COVID mess. The other tool we are thinking about is in insecticidal medicine. So the first picture here is a picture of the cover page of the Lancet. This was June 2018, when we published some of the work we've been doing in Western Kenya, where we were trying to, so ivermectin has been given out for the control of river blindness. And the idea behind it is, if you take ivermectin, if a mosquito bites you, what tends to happen is that your blood becomes toxic to the mosquitoes. And so we're trying to think, how then can we utilize this in controlling mosquitoes? So in this paper here, we did try out a set number of people who are coming to the clinic who are already having malaria. We give them ivermectin in addition to malaria drugs. And every couple of days, we'd get a blood draws from them and feed that to mosquitoes in our insectary. And what we noticed was that all the mosquitoes that we fed on blood on blood from individuals that had been given ivermectin actually died as a result of that, and this went on for almost 30 days. So it means that if you give one person ivermectin, they can kill mosquitoes for up to 30 days. So what happens if you distribute ivermectin in an entire village? Give everybody in that village ivermectin at the same time. Magic, you know. The second one here is the Bohemia trial that is currently funded by United as well, just alongside the special repellence project I just talked about. And this will be looking at giving out ivermectin now to different to humans, giving it to animals, and just trying to see if you actually can derive an impact in terms of reducing the incidence of malaria in populations. The next trial is the attractive targeted sugar-based study. So what you see here is called an ATSB device. And what this is, it takes advantage of the fact that mosquitoes feed on sugar. And so you have these pouches that are laden with a bait that has lots of sugar in it, very attractive to mosquitoes. But the special ingredient that has been added to this menu is a toxin, okay? So different toxins that can be used, of course. And so what then happens is that these baits are hung outside of houses, as you can see in this picture taken from villages in Mali. So then it's hung outside of a wall, and the idea is that mosquitoes coming foraging for sugar sources will bump into this bait, and they'll be like, yummy, this thing smells delicious, tastes delicious. And once they're able to imbibe on the sugar, they'll take in the toxin, and that will kill them, okay? So we're currently doing a lot of work around developing the product further for large-scale field trials actually large-scale randomized control trials that are scheduled to begin in 2022 and end in 2024 in Mali, Zambia, and Kenya. Of course, all these updates will be given at subsequent PAMCA meetings. All right, the next slide shows this is a mosquito swarm. So mosquitoes swarm at the same place at dusk. And so Dr. Diabate's team in Burkina Faso is actually currently evaluating this as a proof of concept. This to try and see if you know where the mosquitoes are swarming, if you come to them with an insecticide and spray them, can you actually spray them in sufficient numbers that you actually end up reducing the number of mosquitoes that are coming to transmit malaria in your population, and therefore bringing down the incidence of malaria. And so this is currently also being evaluated. And then, of course, gene drive technology. So gene drive works in two ways. Gene drive is where you target a region of genomic DNA in the mosquito, and you put in a modification of a gene. And what that modification does, you can either go two ways. One, you can put in a modification that results in population separation. So for example, genetically modified male mosquitoes, you give them a gene that makes all the male offspring of a female are basically, they are unviable, okay? So that if they mate with a female, they're not able to fertilize the eggs. Now what happens, that takes advantage of the fact that female mosquitoes only need to mate once. Once they mate the first time, they fill up the spermatheca and they can't mate again. So for that reason, if they mate with an infertile male, that means that for the rest of their lives, they cannot bring forth viable males. So if you deploy these mosquitoes in a large enough population, you can actually end up suppressing that population. Alternatively, you could alter the population. So for example, if mosquitoes have some genes that allow them to be able to be transmitters of disease, for example, malaria. You could alter that gene so that that gene in that mosquito, it's no longer able to carry the malaria parasite. And so for that reason, your mosquito can bite humans, yes, but if it bites them, it's not able to transmit the parasite. And so if you do this in a large enough population, over a period of time, you can end up altering your population so that your malaria mosquitoes are not able to transmit malaria. And so, so some of the advances in technology in this field are CRISPR-Cas. So CRISPR being what's called a homing endonuclease. Basically, it's a molecular scissors. It can identify specific target sequences of your gene, of your genomic DNA, cut those points, and then insert the target sequence that you have. So Cas is the enzyme that allows it to insert the sequences of interest. So you insert your target sequence, you alter mosquito's you alter that mosquito's genetic makeup, and therefore you're able to input your gene of interest. Now, what happens with gene-derived technology is that, as opposed to normal inheritance, where, for example, if I was to alter the gene and just have it go through normal inheritance, if I have a female that is altered and a male that is not altered, and they mate, so in the first generation, only one of the four offspring is able to adopt that gene. And then if subsequent offsprings will probably undergo gene repair, and it may actually kick out my gene of interest. Now, what gene drive does is that using, for example, CRISPR-Cas technology, same gene that I'm interested in, but now I use gene drive technologies. What it does is that the moment you've put in your gene of interest here, it actually facilitates a repair mechanism where the same gene of interest that has been put in is repaired into the so you know that genes have two alleles. So the repair is done in the next allele as well. So that when you have this now, when you have a gene-driven mosquito versus and a normal mosquito mate, half the population within the first generation will already have the gene drive. Now, if you do that with subsequent populations, you get to a point where within a very short time, all the mosquitoes are adapting to your gene of interest. And so these are currently under a lot of evaluation for possible deployment at a later time. Now, talking about all these tools, and thinking now like entomologists, I just want to talk about now a few metrics that we measure when we want to evaluate an entomological tool. So what do we measure for us to be able to prove that that new tool is actually working against mosquitoes? And so some of the indicators that we monitor are vector density. So you look at mosquito numbers just to figure out, are the mosquito numbers actually going down? You look at sporozoid rates. Now, sporozoid rates basically tell you what proportion of the population actually has the parasite of interest at that time. You look at entomological rate, entomological inoculation rate. Now, the entomological inoculation rate tells you, if you have a mosquito, how many people is it potentially able to transmit the disease to? You look at parity rates. Parity rates are very important because parity is what measures what proportion of mosquitoes have actually laid eggs more than once. And that is an important marker of how old your mosquitoes are. Remember, the older the mosquitoes, the higher the chances that they are transmitting. Because a mosquito can only transmit once it's been able to have a blood meal. So once it's been able to have a blood meal, which usually happens at day 3, so they have to be at least 8 to 9 days old before they can actually transmit. So if you have a population of mosquitoes and your population has mostly older mosquitoes, it means that you have a higher chance of having high malaria incidence in that population. But also it means that your method of control, if it was meant to kill mosquitoes, it is not killing as it's supposed to. So if you have a method of control that is killing mosquitoes, you expect to see a lot of younger mosquitoes as opposed to older mosquitoes. We also look at vector behavior. Some of the tools we deploy. So for example, when you started deploying bed nets, almost all of the mosquitoes were feeding indoors, they were feeding late at night. So when you start deploying bed nets, you suddenly start noticing that mosquitoes are becoming smart. They are choosing to feed in the evening when you're sitting down watching your TV before you go to bed. or they're choosing to bite you outside of the house. And so depending on where mosquitoes decide to bite you, you may need to rethink your strategies for targeting those mosquitoes to be able to control them. Now, talking about vector behavior, vector behavior must be paired with human behavior. Okay, so when you talk about vectors, when I say mosquitoes are biting early in the evening, where are you early in the evening? If we say mosquitoes are biting late in the night, where are you as an individual late at night? Okay, so there are certain human behaviors that predispose people to having higher mosquito bites. So a study by Lydia, I think Lydia Kibe is on this webinar right now. So Lydia Kibe showed that, for example, in coastal communities, they observed higher malaria incidences in people who ride boda. And the reason is these people who ride boda are outside late at night. So mosquitoes have easy access to them. So some human behaviors will predispose them to infectious mosquito bites. Then, of course, we look at insecticide resistance. For example, if you have an insecticide-based control, you want to ensure that your mosquitoes will actually succumb to that insecticide that you're using to control them. And then there are product-specific indicators. For example, if we're looking at partial repellents, I want to measure repellency. If I'm looking at ATSBs, I want to ensure that the toxic sugar beets are actually attractive. They can attract mosquitoes, among other indicators. Now, other efforts that are currently that you currently that are currently being thought about or are currently in place, there are novel chemical formulations that are currently being used for insecticide control products. So some of those insecticides are clonidine, clofenapyr, which are currently being incorporated into either bed nets or IRS formulations. There's combination of interventions. For example, Ben Abongo just published his work on the reduction in malaria incidence in Migori following deployment of IRS in addition to long-lasting nets. There's IRS plus seasonal chemo prevention, and this is work that was done, I think, in Burkina Faso, where they showed that if you're doing IRS, you're spraying the walls of houses, but you also give people medication just around the time when you usually have a peak in transmission. You can actually reduce malaria transmission. There's house modification, so different houses can be modified in different ways. Some of them include blocking the windows, blocking or placing screens on windows, placing screens on the eaves, making sure that doors have screens or other ways that you can prevent mosquitoes from coming into the house. And what we basically see from history is everywhere where malaria has been eliminated, one of the main drivers usually is a change in the design of houses so that people have houses that are mosquito proof, okay? And currently, I saw Lenton on the webinar. So the Division of National Malaria Control National Program, which is basically the equivalent of NMCP in Kenya, proposes a combination of larviciding as well as long-lasting nets, and then long-lasting as well as. So it is beneficial that when you look at vector control, you look at holistic, more things can be done to target mosquitoes that may be out of.
Eric, it looks like we've lost you for that last slide. But you're at the tail end of it. So I think we can end it there, Eric. Eric?
I think he's not able to speak. Yeah.
Yeah, Eric could be having internet issues. So we can move on. Yeah, so thanks to Eric for that exciting presentation, a raft of different technologies and tools that are being explored out there. So before Damaris comes in, I just want to apologize. I'm looking at the time there and it looks like we just have 7 minutes on the clock according to our what we scheduled this webinar to go up to. I would like to request participants that we spill over by just 10 minutes so that we give the speakers at least an opportunity to respond to at least one question each. And then what we'll do is any extra questions, we're going to send them over to the presenters they answer them, and then we can share them through your emails, because we already.
Guys, I'm going to speak, kindly minimize.
Okay, Damaris. Okay, so Damaris is going to speak briefly for 3 minutes about PAMCA, and then we will proceed on to the question and answer session. Damaris, please take it up. Let me unmute you. Okay, yes, Damaris, go ahead.
Thanks, Elijah. I'd like to share briefly about PAMCA, and I'd like to share the screen. So quickly, I know most of us have had or are aware of PAMCA. In full, it's Pan-African Mosquito Control Association. The mission of PAMCA is to provide a platform for African scientists, public health professionals, and other stakeholders to drive efforts towards control and elimination of vector-borne diseases. Our strategic goals include capacity building, whereby people are trained over either specific courses, vector biology, etc., and also to enhance collaboration and partnership within the continent and also outside the continent, beyond Africa. Also to increase knowledge, all manage knowledge, that's knowledge management. The last one being strengthening PAMCA's governance structures. The PAMCA regional hubs include 5 of them. That's we have the North Africa hub, we have the Western African hub, we have the East Africa, Central Africa, and South Africa. Currently, we have about 9 registered country chapters, and they include Burkina Faso, Cameroon, Ghana, Kenya, Mali, Nigeria, South Africa, Tanzania, and Zimbabwe. We'd like to cover the entire region of Africa, so we are looking forward to increasing these numbers. And maybe if you're here and your country is not reflected here, kindly let us know and let us know how we can support you to be able to establish a chapter in your country. At the moment, we have over 250 active members that are not only from research institutions, but we have from the academia, we have implementers, we have people from private sector, and also the NGOs. Currently, the ongoing project includes strengthening vector surveillance systems. That's within the about 9 countries in Africa, and also being able to address the use of genomic data. That's targeting mainly Anopheles mosquitoes. But we desire to do more, because the PAMCA is not only about mosquitoes, but it's targeting vector-borne diseases. So the other activity that we've engaged ourselves in, and you heard about Eric mentioned concerning new tools, is looking at training on gene drive courses. These courses are aimed at providing basic and technical understanding on the tool, that's gene drive, and also to increase public acceptance and regulatory issues and concerning the use of such kind of a tool. Also to equip scientists and public health decision makers and the know-how about gene drive. So far, we've done about 3 courses, and these are done every year. And also we've trained over 75 professionals. The other thing that PAMCA is interested in is increasing capacity of women who are undertaking vector control activities. And we managed to undertake one of the courses where we came together to identify gaps that affect women in their participation in vector control activities. We are enhancing this going forward. The other thing is trying to engage communities. When we get new tools, how do communities get to be involved? So we are trying to develop and test criteria on engaging communities on novel vector control interventions, and specifically those ones that involve mosquito releases, sorry. The other thing PAMCA does is support of the national programs, like the National Malaria Control Programs, and they have done many activities such as larval source management, which has been done in Ghana, Uganda, Rwanda, Tanzania, and Zanzibar. They have been involved in this. The other thing is insecticide resistance management activities, and also vector surveillance and data management. One of our flagship projects that we do every year, and it's been done for the last 6 years, is holding the annual conference. So this, we've had so far about 100 participants attending, and here I'm quoting the one for last year, which was done in Cameroon. But we've done other meetings in Kenya, Tanzania, Nigeria, Burkina Faso, Zimbabwe, and Cameroon. So far, we are targeting, our participants have come from almost 34 countries. What is PAMCA looking forward to do for the next 3 to 5 years? As I said earlier, is looking at strengthening entomological capacity in Africa. The other thing, building and formalizing relationship with national control programs. Also strengthening vector surveillance in the continent. To strengthen collaborations, I mentioned that earlier, and partnerships. To enhance women participation in vector control activity, and this does not only include women participation that are formal employment, but we are going far beyond those people who are professionals to the non-professionals, that's the communities. And also trying to support early career entomologists, and going beyond looking at not only malaria or Anopheles mosquitoes, but also looking at arboviruses and other vector-borne diseases. So what I'd like to add is to join PAMCA in the fight against vector-borne diseases. And today, as we mark the World Mosquito Day, kindly let's be members of PAMCA. Put a link of PAMCA. If you go to this link, you should be able to get information about what is required of you becoming a member, and let's all together fight vector-borne diseases. Thank you.
Thank you, Damaris, for that presentation. So I had requested the participants to give us another 10 minutes so that we can go through a round of questions. So we have 8 minutes to go. So I'd like to ask the presenters, first of all, to thank them all for agreeing to present in this webinar. Now we're going to do one round of questions to each of them, and I would request them to be very brief so that we are done in the remaining 8 minutes. So I will start off with Jeremy. Jeremy, I had a question for you here. So what causes the natural fluctuations in the Microsporidia MB rates within the mosquitoes? Let me see. I hope you can.
Yeah, I think I'm unmuted. Yeah, that's a very interesting question that we are trying to figure out. And the hope is that by understanding what naturally increases the rates, we could artificially manipulate environments somehow to increase the rates of transmission. So to be quick, I don't know the answer to that, but we're trying to figure it out. I believe that it happens when the density of mosquitoes increases after the rains. You then get a burst of transmission, and I think that may be to do with high density of mosquitoes, maybe higher levels or more swarms, mating swarms, in which the symbiont be transmitted from mosquitoes, and then the levels go up. So it's a good question, and I think we're trying to figure it out, but we don't quite have the answer yet.
Thank you, Jeremy. I will have Damaris ask the next question to Eric.
Yes, Eric, thanks for taking us through the new tools. One of the tools that will be of concern is gene drive. And I think despite it being targeted, what is its potential, the impact, ecological impact? What ecological impact will this have in eliminating mosquitoes?
Eric?
Eric? Yeah.
Oh. Okay, let me unmute Eric, sorry. Okay, Eric, you're unmuted.
Great. So in terms of, sorry, just say that again. Let me get your question one more time.
Yeah, what's the potential, ecological potential of gene drive as a tool for eliminating mosquitoes? Because we know that mosquitoes, they play a big role in the ecology also. Okay, in the ecosystem. What is the impact if we eliminated them?
So in the many meetings and trainings I've attended around gene drive, this is usually an issue that comes up. We have a couple of environmental ecologists who not want us to eliminate mosquitoes or the potential for that to happen. But so drive is very targeted. So when we come up with a gene drive acting genetic mosquito, usually it is just that species that it's specific to. So mosquitoes, there are 3,500 species of mosquitoes. One of them, I don't think it will have such an impact that we even notice. It happened that gene drive actually eliminated, for example, Anopheles MB. I don't think that will be a big deal because we're talking about very specific
Okay, thanks, Eric. I think you have issues.
Yeah, I think Eric's network is really bad, but I hope you got a tiny bit of his response. I will ask the last question to Professor Rosemary Sang. So, Professor, thank you for your presentation. My question was, so for the mosquito-borne arboviruses, how much can we get in controlling them with the existing IVM tools that are available to control of malaria vectors, given that some of the malaria vectors are actually also double up as arboviral vectors as well?
Okay, thanks, Juma, for your question.
You can start your video.
Okay.
Yes.
Thanks for your question. Yeah, when we control a vector in an environment where there are agents or disease-causing agents that are circulating that vector could transmit, we will have helped to solve a problem. I guess for Anopheles, that's what I can say. But when you look at the vector species that are causing major diseases like Rift Valley fever, yellow fever, dengue, chikungunya, some of the available interventions like use of insecticide-treated bed nets may not be very effective because of the behavior of the species that transmit those diseases. So I'm not completely dismissing the fact that efforts to control malaria vectors will help us. It will take care of some of the diseases, but maybe not all. And that's why I propose that we need to be more creative to include other species that are important in transmitting arbovirus diseases. Thank you.
Thank you. That's great response. Okay, so we are 1 minute away from the end of this webinar. I just want to take this opportunity to thank you all for, first of all, for allowing us to spill over for an additional 10 minutes. We apologize for that. But thank you so much for making time for the webinar. And we look forward to organizing many more of these and you participating. Like Damaris did present, we request you to sign up to be PAMCA members and be mosquito soldiers from wherever you are. So Damaris, you want to dismiss us now?
Yeah, thanks a lot. Thanks everyone. Looking forward to having you being members of PAMCA. I saw questions asking about us sharing the slides. We'll discuss with the speakers and see how you can get the slides. The last one is, there was a question about certificates. PAMCA management will discuss for those who are interested to get a certificate for attending this session. And thanks a lot for your time. We'll be doing more of this. Kindly, if you like to give a talk or you like to participate in our webinar, reach us on our emails, and then have a lovely afternoon, evening, morning. Thanks a lot for joining PAMCA in celebrating the World Mosquito Day. Bye-bye. Thanks, our speakers. Thanks, everyone. Bye. You should have taken a group picture. Unmute yourself, Elijah. Prof, bye.
I'm trying to remove people.
Yeah, try and remove, and maybe Prof, you can stay. I don't know if our speakers have left. Yeah. Oh, we can hear.
We still have 35.
It's okay. You can sign out. We can restart. Just end for everyone.
Okay. All right, let me do this.